9. So it'll be class 9 is RNA protein interaction, and there's 2 papers here. One by Eva Morfeld. She's from Sweden, Liverpool, and Austria. Like, that's where the 3 people are from, or 3 groups are from. The other paper is by Alex Ariod, and this one's going to talk about protein regulator involved with antibiotic resistance in Staph aureus. So that's those 2 papers. Okay. So the first paper, you see, this is a 1995 paper, so it's fairly old. What I wanted to do with this is show that in molecular biology, there are ways we can look at RNA protein interactions. And this is a good one because this is Staph aureus, RNA3, and the alpha toxin HLA. So Staph aureus is gram-positive. It's a firmicute. It's on skin. And RNA3, we know because of its activity with the CRISPRs.
Yeah.
So it also regulates a whole bunch of Staph aureus genes and also affects the AGR quorum sensing system. So RNA3 acts on quorum sensing as well as regulating CRISPRs. The RNA3 region also has a gene in it, so it's got the delta hemolysin gene. So it's not just in RNAs, it also has a gene inside of it. And it also binds, they know that it binds to the Shine-Dalgarno sequence, and that's how it functions on another protein. The HLA is the alpha toxin, and a lot is known about this because it's fairly important for Staph aureus. It makes pores. Are you familiar with the beta barrel toxins?
No.
No? It's like green fluorescent protein forms a beautiful beta barrel toxin. Well, let's take a look at one. Okay, so I go to Protein Data Bank. And what do we call it? Alpha toxin. And let's go to bacteria. See if we can find one. So in cholera, it forms a pore. There's a pore there. This is Staph aureus. Let's see if we can find Staph aureus alpha toxin. Alpha hemolysin monomer. So this will be the protein.
Can you check Shiga toxin?
Yeah, you can look at Shiga toxin.
Yeah.
See, so right there, I know that this little thing is popping up, but you can see there's a bunch of beta sheets that's forming the pore for the efflux or input of material. And in the case of the Staph aureus, that's a real bad virulence trait, is because it forms these pores. These pores allow leakage and kills the cell. Anyway, so that's the alpha toxin and the pore barrel, beta barrel toxin family. Okay, so in Staph aureus, you've got the AGR locus. The AGR locus is located right next to RNase 3. And within RNase 3 is that HLD gene as well. So this is just a map of the region. Now what they've done is they've taken V8, which is a regular bacteria, and compare that with a mutant where the RNase 3 is deleted. So here, RNase 3 is expressed. On the other side, on the other bacteria, RNase 3 is not expressed. Then if you follow the HLA mRNA, you see RNA3, when RNA3 is there, the HLA is there. When RNA3 is not there, HLA is much in way less amount.
Okay.
Okay, and that's the take-home message from this image. Next, what they do is they show that they're making the RNA3 degrade into very specific products. So if you use the just full-length RNA 3 in lane 1 is a great big smear, and that's if you use non-denaturing conditions. Now if you use denaturing conditions and use the same amount, you get a really nice band. Now in lane 2, what they've done is they've used a restriction enzyme to cut it, and it breaks down to 3 pieces. And lane 3, they're using a different restriction enzyme. So this is a way to take a look at RNase 3. Just to figure out how big it is and if they can just cut it up into nice parts. The next slide, what they have is they show the complex forming between RNase 3 and the untranslated region of the alpha toxin. So if you add all of your RNA, you get these 4 bands showing up. And what they're saying in image B is you can resolve the HLA, or the toxin, the alpha toxin, and RNase 3 by varying what you're adding into the columns. So in figure B, they have again, this is a non-denaturing gel, so it's kind of a big clump. And then in letter C, what they're doing is they're adding restriction enzymes to show that you get different size bands. But you're also getting a complex formed when you add the 2 together.
Where is that?
So here in the control, you get this great big band.
Yeah.
And if you add restriction enzymes, you get...
Yeah, good.
Okay.
Okay.
Now what they're doing in the next slide is they're trying to show that they can figure out where things are. So if you take a look at the top here, RNase T1, primer A4, RNase 3. So in G, that would be your control of... No, sorry. Lane 1 is your control where you're not adding anything. You get some bands forming, which they wouldn't kind of expect. So there is some clumpage or some structure being formed when you're doing in vitro transcription of this
So
region.
it cleaves? Somehow.
It forms some bigger bands than other bands, yeah, automatically. And then lane 2, what they do is they add T1, and you see really definite groups.
Yeah.
Then what they do is they add RNA3. Oh, sorry, no. Lane 3 is adding a primer. And if you add a primer, what happens is you have this region blocked or protected. So it's like they're trying to figure out where the primer is protecting.
Binds to the
region, yeah. And if you add no primer and RNase 3, it's binding there too.
Okay,
so this shows that the
binding.
Primer is binding, or the primer binds the same region as RNA3 binds. And this is the HLA promoter, so, well, just before the promoter, so it shows you that that's what's going on with the interaction between RNase-3 and HLA. Any questions there?
No, not yet.
No? Okay. So the next slide, what they do is they show the sequence of HLA and just show the Shine-Dalgarno region. So there's the Shine-Dalgarno region of HLA, and it's also shown here. And what happens is that region base pairs with itself. So it forms this hairpin loop.
Yeah.
But then what they figured out is, okay, so if this hairpin loop forms and RNase-3 binds, so RNase-3 binds as well at the same time, forming a triple helix in this region. But by doing that, it's also releasing the structure of that hairpin. All right, so...
Okay, so it requires this secondary structure.
Right. Next slide shows the northern blot. I've got the band for RNase-3 showing multiple spots with the northern and HLA showing one fairly obvious spot. Now, there's something weird in this paper. The next picture, so what they do is they take total RNA.
Just a second.
Yep.
So what they are saying here, that RNase-3 binds.
Looks like it's binding something up here. So it's forming these fairly large transcripts, but something is binding up there. And
in the presence of.
If you just probe with RNase-3, that's what it's showing you. And if you're probing with HLA, you get one big band, but that coincides with the RNase-3 band. So what they did in the next slide is they've taken total RNA and do two-dimensional northern blot. So they run it in two directions. So you take the HLA RNA with RNase-3 in one dimension, and then, oops, I've got something over covering my picture. I can go to the other.
It's okay, we have here. Andrew.
This is the Moorfield paper. Oops, here. So in one dimension, they run the gel and then they run it in a second dimension, and what that shows is you can separate the RNase-3 from the HLA.
Okay.
So there's two separation steps. One separation step that way, the second separation step this way. So they've separated the complex, and they're showing what they showed in the last slide. Just
but I knew.
Right.
It's just a
confirmation. Yeah. Next, what they're doing is the HLA mRNA in a mutant without RNase-3. So the K6812-1 is without RNase-3, and if you probe for HLA, nothing.
Okay.
Okay.
So would they want to say that the is the only one biting? What do they want to
on the
conclude?
left here, is
if there isn't.
that they're probing for RNase 3, and RNase 3 shows up here, but not in the mutant. And in the next one, they're probing for HLA. So in HLA, you get a nice band showing up with the complex, but not here. There's nothing, no complex in the mutant.
Which is kind of expected, right?
They were hoping for that result, yeah. Okay, and then the last slide, this is their model. So what happens with HLA is it self-binds. And the self-binding is at the Shine-Delgarno region.
What's this region?
The Shine-Delgarno, that's the...
The cleavage site?
Uh,
Where the RNase 3 binds, right?
well, no. RNase 3 binds on the other side of it.
Oh. Okay.
So RNase 3 binds to the other side of the Shine-Delgarno region, allowing for translation. So when it's in this regular format without RNase 3, there's no translation of this toxin. And when RNase
it's
3
required.
is there,
I see.
yeah, it needs it. So that's how it works.
Yeah.
Yeah.
Wow, in 1995.
They figured out. Yeah, well, RNase 3 was discovered in the 80s. So they knew about it being important, and it just is really important with Staph aureus virulence. So the next paper is...
Yeah, in CRISPR, RNase 3 just helps in processing.
Yeah.
But it's required too. Yeah.
Okay, so this next paper is from EMBO, and this is a bigger
This one,
paper.
Right?
I'm sorry. Oh, NAR. A-Rod, yeah, thanks. And they talk about this region, small RNA called SPRX, and it's called SPRX because it's small pathogenicity RNA-X. They're just naming it by where it's located on the chromosome. Before, what they called it was RSA0R, so they found a long list of RNAs from Staph aureus, and they were just numbering them 0, A, B, C, D, E, F, G, H, and all the way to letter R. So this SPRX comes from another project.
Okay.
The SPOVG is a sporulation gene, and it's important in sporulation for Staph aureus. Okay, so in the first figure, what they show is, on the left-hand side, if you delete SPRX, you don't get a protein, so this is a Western showing no protein.
Okay.
Oh, sorry, Northern showing no... I was thinking this would be protein because there's SPRX with capitals, but it says Northern blot.
Yeah.
So the protein isn't there. Now what they do is they take wild type and they add a plasmid PCN38. It's there, and they add the plasmid with SPRX for an overexpression mutant, and it's really overexpressed.
What's the difference between these two?
So on the WT PCN38 is the wild type with the plasmid.
Oh, okay.
Empty vector.
Yeah.
And
And
then,
still is there.
and the second one is not empty, it's now got the...
They complemented.
Right. Next, what they do is they do an antibiotic susceptibility test, and they just MH as their media, and then they add teicoplanin, and if you take a look, the wild type is very similar to the deletion mutant. In fact, the deletion mutant seems to be a little more resistant to this teicoplanin. And if you take wild type with the plasmid, it's the same as wild type. And if you add the overexpression mutant, it doesn't survive as well.
Okay.
So it implies that by overexpressing it, you're not able to survive antibiotic stress. So it's involved in antibiotic stress somehow.
Okay.
And on the bottom, so they're doing the same thing here with different amounts of the antibiotics. And on the right bottom corner for letter D, this is a total slide. Have you seen these before?
Yeah, they usually do for, I saw it for UV radiation. So this is just the smear, right?
Yeah.
Yeah.
They make a smear, and on the right-hand side, they add your antibiotic.
Yeah.
So you can see if you have it overexpressed, it doesn't survive well. It doesn't.
Yeah, it's just a complementation of what they showed before.
Oh, you had a question?
No.
Oh, okay. So in the next slide, they show you, okay, so SPOVG is in a bicistronic, it has another gene in front of it, and it requires that gene to be expressed.
Okay.
It's in a operon, okay? So if you have this operon working, you get 1,200 nucleotides, and then you can have each gene separated. YABJ is 600 and SPOVG is 500 nucleotides long. Okay.
How did they figure it out? That he...
About what? The operon? About YABJ and...
Why would... Yeah, what would affect the expression of this?
They did an experiment to show that this SPOVG is affected by SPRX.
Oh, okay. So they want to know exactly which component.
Right. And now, if you see, here's letter A. This is your... oops. Making sure. I think this is a Western now.
Here?
Yeah.
A.
A is a Western. So if you add the SPOV protein with the wild type, you get a band. In the deletion mutant, you get a band. Deletion mutant with the plasmid, you get a band. Deletion mutant overexpressed, no band. So it's affecting the protein.
Okay, yeah, I see. Yeah, in the presence of that.
Then if you check the expression of SPRX, notice that you have a band with the wild type, nothing in deletion mutant, but it's overexpressed. Okay, in letter C, they can show the expression of both SPOVG mRNA as well as the complex. And they're doing that in all 4 species.
Okay.
Letter D, they're looking at the operon. When they're together, you get expression of SPOVG. Yeah. And then in the next box, what they're showing is you get the expression is reduced.
How did they do this?
So on the left, this is the operon, and on the right, it's just the gene. So they're showing that the gene is affected by SPRX.
So these, again, it's quantification.
Yeah, this is a real time.
Yeah, no, Northern.
Northern quantification? Oh,
yeah.
I see. Okay.
Northern quantification.
Relative to the wild type. And wild type is 1.
How you can do this?
What you do is you do densitometry. You could use ImageJ, or even
based on the
band intensity,
and
that
band
gives
size.
I see. This gives you these numbers.
Yeah.
as you can see, here's SPO-VG. That's pretty intense there. So that's why they're saying that's close to 1.
Yeah, yeah, yeah.
And if you just look at the single expression of the single region, this is where it drops.
So they just want to, visually it's hard to say.
Yes.
But based on, doing this, which is nice.
Graph is nice, yeah.
Mm-hmm. Wow, yeah. It's significant, actually. You won't say, if you see this, you'll...
Yeah, it's based on the actual expression of the gene. It's not doing the reverse transcription using primers like we would in real time.
Mm-hmm.
Okay, so next what they do is they do this really massive picture of showing its interaction. And the way they do that is they have SPRX sequence, and when they do this, SPRX MUTL3, they're getting rid of a box. They call it the poly-C box.
Where did you get this sequence? Can you get this? There is any software?
3115 to
so
72.
If you want to get this, if you want to get them.
That would just be
convert?
Yeah.
Mm-hmm.
This is just in reverse transcription. So this is just the reverse. Like you take mech vector and reverse that.
Yeah, I see.
And change all the T's to U's.
Yeah.
Right?
I was wondering if there is any soft.
Yeah.
You are aware
mech
of.
vector. Mech vector does that good.
Yeah.
But this picture where it aligns
SPRX
with...
to give you this nice. I think this is...
I don't know how this is done.
This structure.
Yeah, they don't usually tell us in the paper what they are using to make that structure. Because this is a picture I've seen several times before in other papers too. So this is forming a binding complex between SPRX and SPO-VG. And it's binding in this poly-C region. And based on what you see here, so this is around 584, that's in this region there. And that's binding on the Shine-Dalgarno region. All of the arrows indicate cleavage sites, so that's figured out by the different types of treatments. So if it's an open arrow, RNA is V1, that's just giving it moderate cleavage, and you can tell by going up here how it looks. RNA is V1, strong cleavage is a strong band. And they also use nuclease. S1, and there's two kinds of S1 cleavage. You got strong and weak cleavage, and that's also determined.
They made this based on the gel.
Right, and you can see where strong cleavage occurs and where weak sites are. So based on the structural probing, they also add SPRX in this with the control, with S1, V1, lead. I don't recall what G is at the moment, but they're also probing the structure here.
It's this one, right?
RNA is T2. Right.
Yeah.
And then right after GA is RNase U2, and AH is alkaline ladder. So AH is just the ladder following the sequence of the in vitro transcription SPRX.
Okay, so they did this just to get the structure.
To try and figure out where things are cleaving it. And based on the cleavage, you can figure if it's a loop or unattached or bound as part of the hairpin loop. So if it's bound, then it's going to have a different propensity to be cleaved or not. Next, they do the toe print assay, and the toe print assay shows binding of SPO-VG to SPRX. So as you add more SPRX, you get it bound to SPO-VG in this region. And if you add the MUTL3, there's no binding to it. So that loop, that C3 loop, is, or the poly C loop, is really important for the binding.
Okay.
Next one, the figure 6. We've got so they're looking at the C loop again, and if you add the SPO-VG protein in wild type, you get a band, a little bit of a band. In the deletion mutant, you get a stronger band of SPO-VG protein. Then if you add the plasmid, you get a stronger band. And if you overexpress SPRX, no band, it's gone again. But if you add the L3 region, where and delete the poly C region, it binds. So that poly C region is really important for binding. And if you can see in the bottom, SPRX is expressed in both these lanes. So you know it's expressed. It's just that it's slightly different in the structural conformation, and that structure allows it to bind SPO-VG. And in this last slide, what they're doing is they're showing the antibiotic susceptibility again, based on the L3 region. And that's that paper.
They tested this region, huh? They
Right.
say this is. Yeah. Oh, that's nice.
Yeah, okay. Any more questions?
What do you think about this one? Which one?
Well, you can see how far the science has gone from 95 to 2012? No, 2014. That's when it was published.
Yeah.
You can do way more stuff. They're doing much more intricate footprinting now than before.
Yeah.
Before, they're really happy to get it banned.
They love, yeah.
Now they're really teasing apart the details of the structure, like really, really investigating, and that's why it's an NAR paper.
Yeah.
It's because it's so intricate.
But I think this is much. I don't know. I think it's much more better than maybe they had a high chance to get higher impact journal, because they did also the RNA work, but also they did in vivo. So.
And show that it's regulating the glycoprotein interaction. Yeah, resistance.
They did a lot.
Okie doke. I'll just quit this and save. It's Control Shift 2.